Hello, everybody. Welcome to this week's laboratory exercise. So we are doing photosynthesis this week. I am Shannon Reeves, and I am your TA, and here's my email. And this is Professor Ravioli. She is ready to teach us all. So I have to start out with some announcements. First of all, you'll see the same announcements for your photosynthesis lab as you will for cellular respiration, for those of you who are doing it online. And for those of you who are doing it on in person, then just know that you'll get the same announcements, but you'll get them only on the photosynthesis PowerPoint. So this is just because we have 2 weeks that we do things in increments, simply because we have a group A and group B, and these are the announcements I know of right now. So first, grades are in. I've been doing a lot of your grading of your lab summaries. So I encourage you to go ahead and look at all of your submissions. I have left comments there. So keep in mind though, I've only left comments for the macromolecule lab. So if you've got comments on the enzyme lab, then you're lucky. I realized very quickly that this would take so long, simply because of the volume of lab summaries that I have to grade. It would take too long to actually leave comments on everybody's, but I will fill out the portion of the rubric that has to do with what points you've missed. So if you're unfamiliar with the rubric and don't know what I'm talking about, then you're probably not using the rubric for writing. So I really encourage you guys to go into Canvas and look in the rubric for your assignments and actually follow those rubrics, because Dr. Shaw has laid out almost in bullet points what you need to address in each section. So if you're missing points because you're not addressing those things, those little points add up fast. And whenever you have an assignment that's only worth 17 points rather than 21, then it ends up adding up really fast. So just keep that in mind. But because of it, I won't be leaving comments on everything. So if you have any questions about how you did, you're welcome to come see me in office hours or set up a time to talk with me. I'm not going to answer emails about them though, because that's really just doing the exact same thing, but in email form instead of on Canvas. So just so that you know, go ahead and look. And usually the students that do really well in this class are the ones that look back and take all of the critique given and apply it to the future summaries. So be sure and do that. Oh, this is probably the most important announcement, and I'm pretty sure that Ravioli is upset with me because I need to clarify that she does not eat garbage. I got a lot of concerned Flipgrid videos from students telling me that I really should look into getting a trash can with a lid or something that will prevent Ravioli from eating garbage. She doesn't. I just call garbage food. That's my name for junk food. So we feed her protein, but she doesn't eat it often. She would rather have kibble, and I would just prefer that she would eat less grain and more protein because she's a carnivore. But she's the boss, so she doesn't eat garbage though. Just to clarify, I don't need anybody calling PETA on me. Lastly, reaching out for help. Okay, so I am so proud of everybody that is reaching out for help from me. I've had a serious increase from last semester and students that just would like some help, and that's exactly what I'm here for. I would love to help you, but please don't wait until Friday. There is nothing I can do for you on Friday, and it's not just me being like a snarky, mean teacher that's so excited to tell students, no, I can't help you on a Friday. I would love to help you, but I literally have meetings from 8 a.m. until about 4 p.m. So I don't even stop for lunch. I just can't answer emails. So if you've waited that long, unfortunately, I can't help you. But it's a good thing in this class to learn that you probably shouldn't be waiting till the last minute to do assignments that are open all week anyways, and that will help you in your academic career. So just don't try and email. I'm happy to ignore those emails if you've forgotten, but I probably won't even answer the emails if they are asking for help before the assignment is due. So let's look at our lab for this week. You're going to read and complete the laboratory background. So hopefully you've already done that. Now you're watching my video and you're about to watch the simulation video and use the data from that to make graphs. So I will go ahead and say really quick before we get started that the data that you're given this week is different from the data that you've been given for the macromolecule and the enzyme lab. Those two labs, we gave you your exact values that you needed to put on a graph. This week, we're not doing that. We're giving you data that you would get from an experiment like this, and you have to do something to that data. That's called analyzing the data usually. So you're usually performing some sort of calculation, and or you're grouping the data in certain ways so that you can view it with trends. So we're trying to look at the rate of CO2 production. We don't actually give you that rate. You have to find it yourself. So we'll talk a little bit more about that as that comes up, but just keep in mind that that is something that's important that you'll have to do in this lab. Lastly, you'll complete the lab summary and you'll include a graph with that. So just as a reminder, don't include your graph as a separate submission. Canvas won't let you. You have to put your graph in your report in the results section. And lastly, you'll upload it to Canvas and create a Flipgrid video. So it's not mentioned right here, but it's mentioned later in the PowerPoint that Flipgrid videos are due for online assignments. So that will also be due. And actually, before I move on, Flipgrid and any citations that we do actually make up almost a third of your lab summary. So Flipgrid is 4 points and citations are 2. That's 6, depending on how big the actual assignment is. You could be potentially losing 30 points if you're not addressing those things. So just be sure that you're doing all of the elements needed for the week's assignment. Okay, so helpful tips for today's investigation. We are having you guys understand why plants grow better under certain lighting conditions than others. So what does that mean? So I'd like to go ahead and start out with just a little trivia. So what is the largest river on Earth? So if any of you have seen this documentary, it was on Netflix for a while. It's called One Strange Rock. It's narrated by Will Smith, but there's also a lot of other really cool scientists. You have astronauts from the space station that are on there talking about their experience, and a lot of other people that make some really cool observations about the Earth. And one of them they talk about, and I think it might even be the first episode, maybe not, but they talk about the Amazon. And they say, what do you think the biggest river on Earth is? And it is not actually the river that you're thinking of. By the way, this is the hyperlink, or it's not a hyperlink, it's just the URL, I guess. If you wanted to actually go watch it, then that is where you could find it on YouTube, like everything on Earth. I think it's on Disney Plus right now. It might still be on Netflix. But if you wanted to watch this particular clip, I think it's like 6 minutes long. And it's pretty cool. It talks about the biggest river in the world, and it's not what you would think it is. So it is a river that it's existing. It's an invisible river above the Amazon. So it's a river of oxygen and water vapor that is formed whenever the trees go through photosynthesis. When they do that, we know that water is used because water is an input into photosynthesis along with carbon dioxide, which it takes away from the environment to help all of us out. And through the photons that are provided from light energy, it will create oxygen and sugar. So sugar is used in helping the plant, and the oxygen benefits us. So while there's a whole lot of other processes that are going on in order to create this river, one of those is photosynthesis, and it's really a cool video. So I encourage you guys to go look at it if you have an extra 6 minutes of time. But that leads to our calculation for photosynthesis. So you guys are going to be learning about photosynthesis for this next unit in lecture. And you guys may not or may have already had lecture and whenever you watch this video. So you may know that the calculation for photosynthesis is carbon dioxide plus water and a little bit of light creates sugar and oxygen. So the plant will use sugar for ATP, for growth, for creating cell walls, whatever it needs it for. And the oxygen is a byproduct that we humans benefit from. We get to have clean air that we can breathe in. Some of you smarty pants out there may already recognize that this is the opposite of the cellular respiration. So glucose or sugar and oxygen is what we're putting into cellular respiration, and what we get from that is water, because oxygen is our last electron receptor, and then carbon dioxide and a little ATP that we get to use. So back to our lab. Why do some plants grow better under certain lighting conditions than others? Or another way of saying this is, why do wavelengths matter? Why do different colors of light have different effects? So that's what we're going to be experimenting on. So we are going to have a part where we measure the CO2 production, and we take a look at what leaf pigments do and the role that they play in photosynthesis. So your job, though, whenever you're writing in your summary, is to write about how these two things are linked. I don't want to just hear about lights in your lab summary. I want to hear about pigments and what pigments in plants do and how that benefits light and the light that is used by the plant and how that ends up to be plant growth. So we'll talk a little bit about that in just a minute. But this is the plant that you'll do your experimenting on, or that you'll get the data from the experimenting that we've been doing. It's the Schefflera plant, and it's each leaf is about 5 inches long. This is what it's looking like in our chamber. So we have these gas chambers. So as we know, if we have gases, we have learned about passive or simple diffusion. So any gas that's not contained in an area will want to reach equilibrium, and it will want to diffuse out into the air. But we don't want that, because we want to be able to measure the CO2. So we have a chamber that we put the leaf in, and then we cap the chamber, and we put this little CO2 probe in here. So you can see it up in the top left corner. So we put that chamber in the probe into a bucket, and that bucket has LED lights with different colors. So we will be, let's see, sorry, I went too far. We'll be giving you 3 different colors that you can experiment with: blue, green, and red light. So these wavelengths are different, or these colors are different wavelengths that we may hypothesize may have different effects on. You'll also have darkness, and that will be done simply by removing all of the light and placing a paper bag over the top so that there's total darkness in the bucket. And we'll see how photosynthesis occurs there. So I just like to go ahead and ask you guys, we've learned now that we can have positive and negative controls. If we were to have no light, would this be a positive control or a negative control? Would it be something that's responding in the way that we expect it to respond, or would it be something that is a representation of whenever that process does not happen? So Logger Pro, if Ravioli wanted to join this discussion, she might walk over here, but she can't be bothered right now. So this is important. Remember how I always tell you to, Ravioli will pop up when there's something important. So you will be given data, and this data will have 15 minutes worth of data in it. So we're going to stick a probe in the chamber and put that chamber in the bucket. And that CO2 probe is going to be monitoring how much CO2 is being given off by the plant, or I'm sorry, how much is being taken by the plant, because we know CO2 is an input and the output is O2. So that CO2 probe will be recognizing how much CO2 is in the chamber. So for 15 minutes, we're going to be monitoring how much CO2 there is in this chamber. But for the first 5 minutes, there's a little bit going on that we don't really want record of. So we're not actually going to take the first 5 minutes of that data. So all of you will be given all of the data. Do not use all of the data. Use the last 10 minutes of the data, because that's a better representation of what's going on. We will just omit what's going on in the first 5 minutes as if it doesn't happen, but you guys are responsible for making sure that your data represents that. So CO2, this is just a reminder that we have equipment that is used, and whenever we use equipment, we want to make sure we're using it correctly. So a lot of the time we have different settings for equipment, and there have been multiple times when I have done experiments only to realize that I can't do anything with that data because I wasn't using it on the right setting. So this is a reminder to use the low setting for our trial. Okay, so how is CO2, photosynthesis, and plant growth all related? So we have a picture here that shows what our inputs and outputs of photosynthesis are. But as we know that increased CO2 uptake means increased photosynthesis. So if there is more usage of the CO2 by the plant, that means it's photosynthesizing. So that would look like a decrease in CO2. So increased photosynthesis means increased food production for the plant, which they will then use to grow. So increased CO2 uptake means increased plant growth. That's how that is directly related. But we want to remember too that we don't just want the amount of CO2, we want to know about the rate. And if you remember, a rate is over time. So we're going to do a calculation of how much CO2 is being produced over time. So plant cells have mitochondria that are producing CO2 all the time. That's a byproduct of cellular respiration, as we know. If the plant is placed in total darkness, no photosynthesis will occur, and the amount of CO2 in the chamber should increase because it's not using that CO2 for growth. So during the plant, whenever we're during photosynthesis, whenever, I'm going to move this out of the way, sorry. During photosynthesis, the plant will take that CO2 from the air that's in the chamber. So you would see a decrease in CO2 production. So when photosynthesis is occurring, the total net rate of CO2 production in the chamber should be lower than whenever it was during the dark trial. So depending on the color of light that you use in the trial, this plant may still be able to produce more CO2 than it uses. So in other words, really what you want to be doing is comparing each of your light trials to the dark trial. The dark trial is your control that will tell you how much CO2 is normally in the chamber when photosynthesis is not occurring. So that should help you with figuring out your calculation that you need to do. So you can do that over time. That will give you a rate. So if you're doing parts per million per minute, then over 10 minutes, because we're taking the last 10 minutes of our data. So you should be able to find out the rate of CO2 production, and whatever we're trying to find in our experiment should be our dependent variable, just as a reminder. So you're trying to find out the rate of CO2 production in different environments. This environment happens to be different light environments. So why does color of light matter? So you'll learn in lecture that you have light that is used or it's not used. It's either absorbed or it's reflected. So this leaf right here is green. That means it has green pigments inside of it. So those green pigments, the chlorophyll inside of this leaf, are going to use any sunlight that it can. That's its energy source. And this energy source is free. And so it's far more effective of an energy source than cellular respiration. So it's going to try and use as much sunlight as possible, but it can only use the sunlight that's being absorbed. If you have green pigments, you're going to reflect green. That's why you see this green arrow going off of the plant. So that means it cannot use that as a source of energy. That green light is useless to it. So plants absorb some colors of light, but reflect others. More absorption equals more photosynthesis. I have another picture here that kind of illustrates that the wavelengths that are reflected are not absorbed. Those are the ones that we see. So whenever everybody asks, or I was taught as a kid anyways, what color is that object? It's actually everything but that color. And that's kind of a erroneous way of saying that, and a little bit confusing if you're going to rely on that information. So really what that means is you have a red apple right here. All colors of the sun, of wavelengths, are going to be shining on this apple. So it can only use certain wavelengths, so it reflects the red, and that's what we end up seeing. So what if it's blue? Then what is it going to reflect? Blue. But what if we change the light source? So I repurpose this slide in a pretty haphazardly way, but you can see our ball is blue and our light is blue. So we expect that to be the same, but what if we change our light source to be a red light source? If we were to change it to be red light, it would shine on the blue ball. What would the ball absorb? It would absorb the red. So it uses this red light to grow. Or let's say it's not a ball, it's a plant. It's a blue plant, sure. So it will use that red light to grow. What's being reflected then? Nothing. Nothing gets reflected. We see black. We don't see any light because all of the light's being used there. So that should help more in lecture than it will in the lab, but the absorption part should totally help you in this lab. So you need to make sure that your items, your plants that are being shown light onto them can use the light that is being shined on them. So what pigments do our plants have? I'll move this guy again. So our plant specifically has these three pigments: chlorophyll A, B, and carotenoids. So leaves contain pigments or molecules that absorb energy from light. These are the three molecules that absorb that energy. So chlorophyll A has this green, darker green color. Chlorophyll B kind of has this orangey green color. And then this yellow carotenoid is the third pigment. It expresses as a bright green, as you can see, but those are all the pigments present. So since it presents itself as a darker green, a bright green, then you might expect that not all of these pigments are in there at the same amount. So maybe not all of them are good sources for getting light. So in this plant, we see the carotenoids will take in a whole lot of blue light. So this is our wavelength on our x-axis, and it's saying that these are the different color options, and this is the absorption percent. So whenever we have blue and violet, then carotenoids tend to absorb more of the blue, maybe even teal color, and less of the violet. No orange, no red. So the chlorophyll A absorbs a whole lot of this blue area, even more so, but less of the teal area, very little green, yellow, but then we get over to orange and red, and again, we have another spike. So it can use a lot of those wavelengths. Lastly, we have chlorophyll B, which gains the most from this blue light and quite a bit, again, from the orange part of the spectrum. So this is advantageous for a leaf because it doesn't just have one molecule that will start taking in photons as energy. It has three different ones that kind of do different jobs. It's kind of like a car that sometimes runs on gas but can sometimes be electric. Now you just have two sources of fuel there for you. So given these pigments that are in there and this graph that tells us the different types of colors or wavelengths really of light that are being absorbed, you'll develop a hypothesis about which wavelengths you'll think will be the most effective at photosynthesis. So your hypothesis, I will again move this just a little bit. So your hypothesis should link light color to plant pigment and photosynthetic rate. So if your hypothesis just talks about pigments, it will not be enough, and that will not be a point for hypothesis in your introduction. If it just talks about wavelength and plant pigment, it's not enough. A good hypothesis will say something like, plants photosynthesize faster or slower under blank conditions because, and your causal statement will have something to do about the pigments that you observe in that particular plant. So based on your data, can you recommend a specific light color that is best for plant growth? And if you can, you'll probably want to talk about this in your conclusion. So another thing that we're adding that we would like you guys to include at either the beginning or the end of your summary. It doesn't need to be special. It doesn't need to have its own section, and it won't count towards your word count. But I need you to identify what your hypothesis is. You'll still include this in your lab summary. Your independent variable and your dependent variable, still include this in your lab summary, and your control and experimental group. So I'll go ahead and say the number one thing that I counted off for the lab summaries that I have graded are that the methods section has yet to identify your control group and experimental groups. Some people will maybe mention a control or that you had a positive and a negative control, but won't actually identify them. Those things need to be identified in your methods section. Hypothesis needs to be identified in your introduction. These 5 things need to be also included either before or at the end of your summary. I would prefer before, just as little bullet points. I don't care how it looks. I am not worried about that. And again, like I said, it doesn't count towards your word count, but I do need you to identify them. So lastly, reach out to me if you have questions. Ravioli's here to help as well. And I am here to help. But again, the best time to be able to contact me is not on a Friday. I would be of much more help before then. So we do have citation stuff for you to do this week, and it will be a part of your grade. So finding and citing scholarly sources, you're going to, again, find 2. I didn't delete that note, not a huge deal. Go ahead and cite 2 scholarly articles. I want you to do the first 2. So I didn't really make that requirement last time you had to do citations, but I want you guys to do this next time or this time. So I want you to actually do the top 2 citations that you find whenever you look for the terms photosynthesis and biofuel. Now keep in mind that you need to change the title. So that just says to correctly cite it, and you'll want to look at your how to cite in Canvas in order to do this. Okay, so this is what Web of Science looks like for you guys. So photosynthesis, you'll want to put this and biofuel in your basic search, but over here, whenever you it asks you where you want to see these. So it could be just something that's in the paper, or it could be in, you might be searching for authors' names. So we want to see photosynthesis, add a row, biofuel. Those are both going to be in the title. That should get you your top 2 sources that you need to include in your literature cited. Again, you don't need to include these in your paper this week. So we just want you guys getting better at citing and being able to properly cite in your literature cited section. So tips for success: review the how to write lab summary, how to write a lab summary document in the lab resources module, and review the assignment rubric before writing or during. I usually encourage students to have it out in class while they're writing. Your lab summary should be uploaded to Canvas by this Friday at 5 p.m. And your Flipgrid post should address the following in 1 minute. So I just went ahead and put all of that stuff on Flipgrid. That way there's no confusion. I think last week I put different things in different places. So if anybody has questions, again, I'm here to help. And feel free to email me or set up time to go over things either over Zoom, or we can do things in person. So have a great week, and I will see you guys later.